Biochemistry and Cell Organisation Taskbook. Question 3. Cell structure and organisation. Name structures A to D and give one function of each. There's A there. So there could be some slight confusion with A. But I think we're sure it's going to be the cell wall there. It's pointing to quite a thick part of this. I mean, the confusion could be possibly membrane, but I can't see any cell membrane, and it tells us it's a plant cell. So we can go for cell wall. Function is there for support, turgidity, stop the cell from bursting, that sort of thing. So let's go for one of those. Stops cell from bursting. Or let's go for turgidity as an alternative there. Maintains the shape of the cell. B. Some slight confusion could possibly arise here because does it look like a mitochondrion or a chloroplast? And it's not in colour, so that would help us. I can see some other little objects down here that look a little bit like mitochondria. And here in the chloroplast, I'm looking for a double membrane, can't see that, but I can see something there. That could be a starch grain. I can see thylakoids and stacks called grana. So I'm going to go for chloroplast. And then function, we're going to have a photosynthesis, or you could indeed put a description of photosynthesis down there. C, some confusion, possibly it's one of the harder things to remember, but this looks like where the cell wall is reduced and there's some sort of a gap in the cell wall. So this is a plasmodesma, or plural plasmodesmata. So plasmodesmata. And we can just say here movement of materials between cells, cytoplasmic connections between cells. So we can say that movement of cytoplasm between cells. D, I think it's referring to this entire structure here. So vacuole, and again we can be quite general here, storage of materials or storage of sap. Over the page. State concisely, which means not war and peace, just give us a brief overview of how each of these structures will be involved in the process of protein synthesis, so making proteins, and the job has got to be to do that. So mitochondria, we might be tempted to say these have actually got ribosomes in them that might make proteins, but it's involved in it's given us a big general plant cell there. So how are proteins made in this, and mitochondria are there to provide ATP energy for this, so to provide ATP energy in aerobic respiration. Ribosomes, well, these are the actual structures that join the amino acids together, form peptide bonds to make the protein. So let's say that join amino acids together, form peptide bonds. And later in the course you'll discover about messenger RNA and the code being read by ribosomes. Endoplasmic reticulum, We'll note it doesn't actually say rough endoplasmic reticulum, smooth endoplasmic reticulum, so we can't just say protein synthesis here. We need to say something possibly to do with the ribosomes could be attached to it, or it's involved in proteins being secreted onto it, or moving, or vesicles, that sort of thing. So we could have ribosomes are attached to it, or possibly formation of vesicles. These vesicles would then move to this Golgi apparatus, or Golgi body, and if you ever want to say one thing about Golgi, it's where proteins will be modified. So modification of proteins. Quite often the quaternary structure of protein gets turned into a glycoprotein, that sort of thing. But we don't need to go into detail. Cell surface membrane. Well, this protein might be taken out of the cell, so we just need to really just name the process or a description of it. Here's the process: exocytosis, or movement of the protein through the membrane and out of the cell. Nucleus. Well, this is where the code for the protein originally is. So we can say the code for the primary structure is on DNA, and that's in the nucleus. The nucleolus, well, that hasn't got the code for the protein on DNA. That's involved in making something else useful. So here we are, making ribosomes. Or what are ribosomes made of? They're made of rRNA. So makes ribosomes, or ribosomal RNA. An amino acid was taken up by the cell and incorporated into a protein that was secreted from the cell. The route can be taken by a flow diagram of cell structures. So this is amino acids being taken up and then made into proteins and then out again. So the amino acids would never actually come into contact with the nucleus, but they get turned into a protein using these above. So from the list in B, so amino acids wouldn't be anything to do with these. So where would they first get joined? Well, that is in the ribosomes. Then they get transported through the endoplasmic reticulum. And then with the Golgi body there, where they might get modified. I'd love to say the membrane here, but well, I can't say the membrane because it tells us it's the membrane there, so we're not going to say the membrane. But he named the carbohydrates, so whatever we say, it's got to be carbohydrates, monosaccharides, disaccharides, polysaccharides, in the cell opposite that I found in the cell wall. So hopefully you can remember cellulose being in cell walls. And then storage compound in the cell. Well, we've got two polysaccharides that are used for storage. Remember, monosaccharides and disaccharides are soluble and will interfere with the osmotic potential of the cell, so they're not used. So we're looking for a polysaccharide storage. It's either got to be starch or glycogen, but which one's in plants? Well, that's starch. And then a little trick question on the end is a component of ribosomes. Well, when you do nucleic acids, we'll do the structure of RNA. So ribosomes are made out of RNA. What's the R in RNA? It's ribose, which is a pentose sugar. And that's 16 marks.